Hello students and welcome. This is a lecture on leaves, form and structure. I am Professor Fernetovich and I'm here to guide you through this topic. So we've talked about some of the other organs in plants. We've talked about stems and roots, but now the last organ that we're going to talk about that's vegetative are the leaves. These vegetative organs are parts of the growing plant and not part of the reproductive structures. The functions of leaves are mainly to collect solar energy. So they are the main site of photosynthesis for most plants. They also allow for CO2 to be absorbed by the plants because they have small pores in them called stomata. We're going to see some pictures of that. There are many adaptations for leaves. I talked about one in the Moodle site, pine needles, but at the end of this lecture you will see some other leaf adaptations with specialized functions. On the right we have a picture of a plant highlighting the leaf right here. So leaves as a whole are where photosynthesis takes place. However, cacti are a big exception to this. The cacti leaves are the spines that come off it. So those thorns that we think of as the prickly parts of a cactus, those are actually modified leaves. And you can guess what that modification is for. It's actually an adaptation to prevent herbivory. Keeps insects and animals from chomping on the plant. And the big green parts of the cactus are stems. A leaf is generally made of a petiole and a blade. So that little leaf stem has a technical name to it called the petiole. Kind of sounds like petal, but it's different. And the leaf blade is the part that we usually think of as the leaf itself. There's a lot of variation with leaves, but there are 2 or 3 major arrangement patterns that we see. The opposite leaves occur where 2 leaves come out at one node on opposite sides of the stem. Maple trees have this type of arrangement, ash trees. There are a variety of trees and plants that do this. Alternate is actually more common. That's where we have 1 leaf per node, where they're staggered. We've seen some examples of these opposite and alternate leaf arrangements in our local plants. Whorled is something we haven't seen yet. This is where 3 or more leaves come out at a node, and they form this circle called a whorl around the node. And it could be a very simple one like the catalpa here, or it can be a more elaborate whorl where there are many leaves coming out at the node. You can also see this in branching patterns. An example of a tree with a branching pattern of whorled is the pine tree. If you take a look at pines, you'll see branches come out all different sides of the trunk, and then you'll see the internode, and then you'll see another round of branches. And you could actually count those whorls up the tree and get an estimate of how old the tree is. That's an alternative to cutting down the tree and counting the rings. You can actually count the whorls and determine an average age for the tree. Monocots and dicots differ in the arrangement of their veins, and we've seen this previously in class. I've talked a little bit about this. Monocots are things like our grasses, and dicots are plants with more broad leaves. So monocots have parallel leaf veins and longer, slender blades. This looks just like a blade of grass up close. Lilies also fall in this category. Dicots have branch-like leaf veins. The leaves is more palmate in shape, broader leaf. We did see the internal structural differences in monocot and dicot stems and roots, The internal structure of leaves is fairly similar when we look at the arrangement of tissues. Each part of a leaf has an important job. This is showing a cross-section of a leaf. So imagine this is like a sandwich where the inside of the sandwich consists of this mesophyll and the vascular bundles right here. So on the uppermost side of the leaf and on the bottommost side of the leaf, we typically have a cuticle. This is a waxy layer that's excreted by the upper and lower epidermis. So the cuticle can be very thick, depending on the plant, or thin. Thick cuticles are characteristic of plants that live in dry environments. Below the upper epidermis, we have palisade mesophyll cells. Meso means middle and phyll means leaf. So these are middle of the leaf cells. And they're kind of squiggly in shape here. They can vary in shape, but they are kind of typically columnar, if we can imagine that, the column shaped. They have chloroplasts in them. Okay, so that's a site of photosynthesis. Notice that the upper and lower epidermal cells do not have chloroplasts. These odd-shaped cells, also in the middle here, are spongy mesophyll cells. These are a little amorphous without a distinctive shape, and there's usually some space in between them. It's dramatized here, but that allows for gases to pass through the leaf. Spongy mesophyll cells also have chloroplasts and do photosynthesis. But because they're below the palisade mesophyll cells, they do a little bit less, and that's why they have fewer chloroplasts. This is a vascular bundle, as is this. And when they're in leaves, we call them leaf veins. But same type of vascular cells that we've seen before, xylem and phloem, surrounded by a bundle sheath cell. I'd like to point out down at the bottom of the leaf in the lower epidermis, we have a guard cell right here and a guard cell here and a stoma right in between. One is shown over here as well. This is a tiny pore that allows for gas exchange into and out of the spongy mesophyll cells. So the stoma is where the CO2 goes into the leaf and where oxygen is released from the leaf. Here we can see the stoma shown a little bit more distinctly. Right here in the guard cells. I want you to think about why they are on the bottom of the leaf, not on the top. Okay, you can pause it if you want to think a little bit harder on that. I will let you know that the stoma are kind of a vulnerable place for the leaf in that organisms can go into it, they can invade, and I've seen fungal hyphae go into stoma to exploit the leaf. But most importantly, the stoma can allow for evaporation of water. And so when the stoma on the bottom of the leaf, water evaporation is a lot less common. If there were stoma on the top, water would easily evaporate. But down here, it's more difficult because the water vapor doesn't travel downward, it would go up. This is showing a typical cross-section of what we would see under a microscope. So this is pretty common. We have some vascular bundles here and here. They look different because one is a cross-section and one is more of a longitudinal section. If you could imagine the branching of the leaf veins, you're going to have different angles from your cut here. So that's why this looks a little different than this. Guard cells and stoma are shown here, the spongy mesophyll with all its space, and the palisade mesophyll above. It's also called palisade parenchyma. It is ground tissue. So we have, of course, ground tissue, dermal tissue, and vascular tissue are three main tissue types in the cell, or in the plant. And a monocot leaf looks fairly similar too. Big vascular bundle here. You could always tell because we have thicker walls on these cells and our big xylem cells. Epidermis, stoma, bundle sheath around a vascular bundle. This is a big mid-vein and smaller veins going up along the sides. Transpiration is a special form of evaporation that's done by plants. So plants, I've mentioned before, are kind of like giant straws, sucking up water from the soil and evaporating it out the top. And this causes a vacuum through the plant. And water is also moved up from the roots to the stems, to the leaves by capillary action. That is the attraction of water to the sides of the xylem and phloem. So it's attracted to the sides and also pulled up. So most of the water that plants take up is lost to the atmosphere by transpiration. It's a big part of the water cycle that's often not discussed. The rate of transpiration is controlled by the stomates, or the stomata. Okay, stomata is plural for stoma, and here we have the guard cells with the stoma right here. Stoma can open and close like this, like little mouths. And depending on the plant, it will do that in various situations. Some plants will close their guard cells during the day when evaporation rates would be high to conserve water, and they'll just open them at night. Others are open various times during the day. Some will open when it's raining out, some will close when it's raining out. But those guard cells can change and allow for gas exchange. Okay, I've got some specialized leaves here. So we talked about the cactus. Okay. We have some interesting leaves here. And in the Moodle site, I discussed pine trees, where the leaves are the needles, and they're adapted to living in dry and cold environments. So water is not available during significant portions of the year in very cold environments. It's got to be liquid for plants to be able to absorb it. And so if we have ice or snow, that's not available water for the plant. So these plants have the adaptations of needles to survive that. They've got sunken stomata, thick cuticles, needle-like leaves, which also shed the snow easily, and a hypodermis, which is an extra layer of cells just under the epidermis. Cotyledons are the first leaves of a germinating plant. You've seen these when you started your plants at home. These are different types of leaves, and they store food for the plant when the plant is in the seed. This is a cross-section of a corn kernel showing a cotyledon right here. You've learned about pitcher plants as well, and this is a modified leaf. You can actually see the change of the pitcher plant leaves in the greenhouse over time. You can see the little leaf come out, and when it needs to acquire that nutrition from the insects, it will form this cup. Very cool. There are some great places to see pitcher plants around us. One is Holly Bog in Holly, Massachusetts, and I definitely recommend anybody go there and view the plants. Please don't pick them though. The Venus flytrap is another specialized leaf, and our last one of the day. Turgor pressure and ion movement of the leaf cells cause the trap to shut. So it's sensitive to when an insect touches it, and then the leaves will snap together, trapping the insect. Sometimes it's fast, but I've seen this happen quite slowly, and a fly, unless it's kind of stuck there, can escape. That is what I'd like to tell you about leaves today. Please email me if you've got any questions. Thank you.